Our next speaker is Ritchie Ho from Cedars Sinai, and he's going to tell us about CRISPRing repeat expansions.
Good afternoon. Brief background about myself. I'm native Los Angelino, born and raised here. I did my undergrad up north in California at Berkeley, and then I just boomeranged back down here, did my PhD in iPS reprogramming at UCLA, and then wanted to continue my passion for disease modeling with iPS cells, and so I went to Cedars Sinai to do my postdoctoral work. And since 2020, I've stayed as an independent PI. So that's a little bit of my background. And up until now, I've been more disease modeling, but I've had some brilliant trainees come through the lab, and they have great ideas that culminated into a gene therapy grant, so which is why I'm here in this session today. So this is a talk focused on targeting repeat expansion diseases. And I'll tell you a little bit about the challenges that characterize this class of diseases and some of the ideas that we're employing with CRISPR-I as a gene therapy approach. So this is a timeline, a little bit of history on the discovery of repeat expansion diseases. They all, by and large, culminate into neurological symptoms that have variable outcomes, either learning disabilities or motor disabilities and other sorts. But the characterization of the genetic forms started in 1990, and this is through this linkage golden age. But given more contemporary technologies, we're able to discover more with like long read sequencing, which these are nucleotide repeat expansions that span thousands of base pairs in the genome, so they're really difficult to detect. But some of the more well-known ones you may have heard of are Fragile X syndrome, Huntington's disease caused by these CAG repeats in the coding region of the Huntington gene, Friedreich's ataxia, and CNR north frontal temporal dementia, ALS, which is the focus of what my lab studies. So structurally, what occurs is you have these trinucleotide, 4-nucleotide, and 6-nucleotide, n number of repeat expansions that occur and disrupt the gene body at different places. You can have it disrupted in the exonal regions, and these are protein coding regions that can give rise to these toxic peptide repeats. But you can also have these repeat expansions in the promoter regions that are in the 5' or 3' UTR, as well as in intronic regions, and these can disrupt regulatory elements within the gene body. So the tricky thing about a lot of these expansions is they cause pleiotropic effects. On the left side, we see a figure illustrating the normal processing of mRNA into protein, but when you have these repeat expansions, say in the promoter region, you can have, depending on the nature of the nucleotide, CG repeats can cause aberrant DNA methylation and disrupt regulatory control over the expression. On the bottom, you can also have RNA structures that can form naturally, hairpin structures that can form from a healthy level of repeats, but when they get to a larger size, they form aberrant tertiary structures in the RNA, and that also disrupts expression, as well as disruptions to RNA splicing. And then these are known as loss of function effects, but at the same time, they can also cause gain of function effects by introducing, say, those coding region peptide repeats that can produce proteins that form aggregates that accumulate in the cell and disrupt normal cellular processes. And it doesn't have to be in the coding region. When you have repeat expansions in the intron or upstream, you have an event called RNA translation, which is non-canonical translation, not requiring an ATG. And so you also have the production of these aberrant peptides that form and also form aggregates. So I think a characteristic of these diseases is one, the detection challenge, because these repeat expansions across generation and maybe even across cell cycles, they can expand and contract. And so in an asymptomatic patient where repeat expansion undergoes expansion to the pathogenic length, offspring of these individuals can present with these diseases. And so it makes it a challenge in anticipating it. Now, due to this feature, there are treatment challenges associated with it. So as I mentioned, there are pleiotropic effects, both loss and gain of functions. So how do you know which function to target if you were to develop one therapy? And the variable repeat length can vary across patients and even within the same gene. So for instance, FMR1, which is the repeat expansion that causes Friedreich ataxia, that's within a certain range, like 50 to 200. But when you get expansions above that, then you have Fragile X syndrome. So the presentation of diseases varies based on the disease length. And so across patients, this can vary, but also there's a concept of more recently appreciated of mosaicism. And what I mean by that is, for instance, this is the myotonic dystrophy gene that on the left-hand side, if you use long-read sequencing, you can detect two distributions, right, of two alleles. And these are very, defined lengths in this patient sample. But in a patient that has repeated CTG repeats in this gene, on the 0 side, you have basically no repeats. You get sequencing reads that indicate no repeat. But on the other allele, clearly you have a range of repeats. So if every different cell in the body has different repeats, how do you deal with that? And how do you come up with a gene therapy that can really target all of them potentially, or make a significant modification to the activity of some of them? Oh, sorry. So on the right-hand side, we have some attempted gene therapies. Most popular, in my opinion, that have come through the clinic is antisense oligos, but also along the lines of RNA inhibition, right, where you design gene therapies that target and silence the repeat expansions. But in many instances, it's tricky how you can control which allele you can properly target. So our main disease that we're focusing on in our lab and working on this CIRM-funded project to develop a therapy for is C9orf hexanucleotide repeat expansion. So this is the most common form of familial ALS, so a brief background on amyotrophic lateral sclerosis, or Lou Gehrig's disease. It's the specific degeneration of cortical and spinal motor neurons. And when patients first get symptoms, they typically die within 3 years of their onset. So it's a very rapid progressing disease that has currently no cure, save for some drugs that extend lifespan by 3 to 6 months. So there's a clear unmet need for this patient population. Now, only 10% of them are familial, which contract to about 50 mutations. And among these, the largest majority of them are these hexanucleotide repeat expansions in the C9orf gene. So this is the gene body of C9orf, and normally the hexanucleotide repeat expansion is shown in green, and it's found in the first intron between alternative exons A1A and 1B. And when a normal healthy allele would be between 2 and 24 copies of this 4G and 2C repeat. And this, you have several splice variants of it that use alternative exons, exons. Now, when you attain a pathological repeat expansion between 30 and upwards of thousands, you get gain of function toxicity in the production of these RNA foci. Right? They form quadruplexes when they contain all these repeats in the RNA molecule, and they form aggregates in the cell. Similarly, you can also produce these non-canonical translation products known as dipeptide repeats, and so these are also shown to be toxic for the cells, and these are primarily characterized in neurons. But an interesting component to this is that the expression of the C9orf gene, which is involved with endosome trafficking, you suffer a loss of function. So having repeat expansion in one allele causes a drop in expression, and this is truly felt more so in the immune compartment, namely the microglia, which undergo a hyperinflammatory tone. So you have both gain of function and loss of function. So how do you develop a therapy that can silence the gain of function but ideally aim to not further repress the healthy wild-type allele any further than it should be. So some candidates have made it to clinical trials, but unfortunately they have failed and not moved past phase 1 and phase 1b/2a. So these were from 2 companies, Biogen and Wave Life Sciences. And this was ASO, antisense oligonucleotide product that was meant to target regions flanking the repeat expansion, right, shown in the lower left-hand diagram. And while this demonstrated really good targeting engagement in the animal models, basically under these phase 1 trials, the patients and the participants didn't feel any meaningful slowing in the progression of their disease. So these were halted. So that led us to wonder, why have these therapies failed, and could it be, 2 possible hypotheses, right? The design of a very specific antisense oligo sequence could target not the repeat expansion, but the areas flanking it to a very specific region that's common between the disease allele and also the wild-type allele. So it could be a non-allele specific targeting. So you could, probably target the wild-type allele in a bad manner. Or just a lack of target engagement in a subset of patients, say for instance, patients with extremely large thousands of repeat lengths, maybe that one product was not able to effectively engage a target in those patients. So postdoc in my lab, Rajesh Singh, who's also a CIRM trainee, and he's presenting a poster, but not on this, and another project that he's working on. But he comes from training in CRISPR therapies, and so he came up with this quirky idea to use CRISPR-Cas9, CRISPR-I, which is CRISPR interference. So this is a dead Cas9 that does not introduce DNA cutting, but rather fused to transcriptional suppressors in the form of heterochromatin establishers. And so you have your guide RNA sequence to guide the complex to your target gene, in this instance C9. And this would establish chromatin marks to help silence the gene. But in panel B, you would see conceptually that if you had the perfect guide RNA sequence guiding to the repeat expansion, it would target the complex to both the repeat expansion allele on top and also the wild-type allele on the bottom, and that would lead to strong interactions and no transcription of both. So we don't want that, right? But the idea is to modify either the length of the guide RNA in this instance so that you'd have less affinity for your repeat expansion sequence. And so when you tinker with the ability of this complex to target efficiently both alleles, the fact that there's much more landing sites on the repeat allele would enable, by probability, a better chance for this complex to seed heterochromatin and therefore silence only the expanded allele, but not strong enough to bind to the healthy allele and therefore basically leave expression of the healthy allele unperturbed. So he developed a screening system. I'm not sure why I was afraid of this, but okay, so the Cas9 is just an image of the dCas9. So he initially set up this assay with a HEC293 T cells, where he has the dCas9 transfected in conjunction with these guide RNAs targeting the repeat expansion with variable lengths from 20 to 10 and 14 in between. And then also a series of allelic reporters, so going from 3 repeats of this GGC GFP reporter all the way to 99 repeats. And so that's meant to reflect the disease allele. And the goal is to see whether modulating the size of the guide RNAs would effectively suppress the disease allele, but not repress the healthy, shorter repeat allele. And so this is quantified by just measuring and counting the number of cells that have been transfected with this GFP reporter. But in panel B, this is to demonstrate that the CRAB9 and the MACP fusion domains are necessary for this repression, whereas in the blue data points are a non-targeting guide RNA, no repression. You do get about 50% repression when you include the full dCas9 CRAB MACP. So he also demonstrated with the system a dose-response curve, which is important in developing a therapy. And so the more guide RNA you deliver and transfect, the stronger its repression effects. And so over on the right-hand side, you'd see that 100 nanograms of guide RNA delivered, whether it was the 10 nucleotide long or the 20 nucleotide long, efficiently suppressed the long allele. But if you look at the same concentration in the shorter reporter, the 16 GFP at 100 nanograms, you see that there's a specific ability of the 10 nucleotide long guide RNA to suppress only the long allele, but leave the shorter allele unperturbed. Whereas with the full-length 20 nucleotide, you get the regression of the healthy allele, which is demonstrating this, the efficacy of this length modulation. Okay. So, so then this is a further demonstration of a different series of reporter alleles. So going from 3 repeats GFP all the way to 16 to 33 to 99. And the key contrast I want to draw here is that at the 99 repeat on the far, your right-hand side, the 10 nucleotide long efficiently suppresses the 99 reporter, right? And then back over to me, on the left-hand side, the gray bars would indicate that this would more or less leave the wild-type allele intact, right? And so this is not the case for the longer nucleotides. So demonstrating, again, another principle that, you know, the length of the guide RNA can affect the efficacy of the suppression. And then finally, another element is not just the length of the guide RNA, but also the sequence. So, you know, you reduce the affinity of the guide RNA binding to its target by reducing the length and the number of hydrogen bonds. But also, you can also actively introduce point mutations of varying degrees, and this is shown in the red highlighted nucleotides in this panel, where Brajesh mutated selectively different regions and at different positions and a different number of nucleotides, right? To demonstrate that you can modulate the specificity of the suppression by not targeting length, but also the actual sequence of the guide RNA. And so the one standout guide RNA is short guide RNA number 3, which demonstrated very effective suppression of the 99 allele, but left the 16 expansion allele unaffected. So these were basically the principal data that enabled us to secure a DIS2 grant. And so we're now 6 months into it, and I don't have new data to show you yet. So, but in summary, repeat expansion disorders disrupt gene function, and they also at the same time can introduce toxic molecules. So this remains a challenging feature of these diseases to develop a gene therapy for. And so our hope is that by tinkering around with these biochemical properties of the guide RNAs, we can have a broad range of patients that we can treat, right, with one product. And in principle, hopefully, the more repeat expansions you have, the efficacy of this drug could scale with the patient's genotype. Yeah, so I guess just to pitch our project and just to describe what's to come. We have 4 major arms that we're working on. First is developing a dual reporter screening platform. So I showed you just the GFP as a reporter for both alleles, but we want to do a simultaneous red and green reporter that both reporters represent one long allele and the short allele, establish a system, and then upon the system, we're going to do a more comprehensive screening of guide RNAs through a library screening method. We'll do lentiviral transduction, sort out, the evenly expressed alleles would be yellow essentially, but we'd be screening out just specific repression of one fluorophore, and then use next generation sequencing to look at the barcodes of enriched guide RNAs in those populations. And this is again just an image of the systematic deletions and point mutations we'll be introducing. So it's about 1,500 guides. And 3, we're going to leverage human iPS models of motor neurons and microglia. So these are ALS patients that carry the C9orf genotype and of varying lengths. And so we want to see target engagement as well as disease phenotype reversals in this model platform. And so we'll be differentiating these iPS lines into motor neurons or microglial cells and delivering the guide RNA that we've learned from the screen and to observe a reversal of some of the RNA foci and dipeptide repeats that we can see in these models. And then we hope to verify specificity by doing single-strand transcriptomics to look for off-target effects. This is just an illustration of the panel of patient lines that we're attempting, and I refer to this as a clinical trial in a dish, in that we really want to represent a patient population, not just both in terms of male, female, but also a range of different allele sizes, and so that's shown in the lower right-hand graph between male and female control or male-female C9 lines with a range of different repeat expansions, and we hope to see, test our hypothesis that the suppression of this disease allele can scale with the repeat expansion length. And then finally, we're also working on demonstrating in vivo efficacy with transgenic animal models, mouse models of this C9 repeat expansion, and optimizing the AAV serotype that we need to use in order to infect the blood-brain, the brain. And then we're testing a panel of AAV serotypes because this has been challenging to find the correct AAV vehicle that can work in mice, but also in primates, can cross the blood-brain barrier, work in cell culture models. We included AAV9 as a positive control because this has been a FDA-approved treatment for SMA. But we hope to find a way to deliver this systemically without having much tropism for the liver. So I think, I'm over. But credit to Brajesh, my postdoc, and my team, and all our funding sources. Thank you.
So the question is not specific to your project, but how do these mutations come about? And especially, how does mosaicism happen within the patient? So one of the things I've kind of figured is that the CAG repeats expand when DNA replication is happening, and it seems to be on the shorter Okazaki fragment part of the DNA polymerase, but I'm not so sure about that. And then to kind of I always thought that these were gametes that were mutated and that inheritance of that affects the genotype. But you mentioned that there are variation within the patient as well.
Yeah, so I'm agnostic as to a unifying mechanism that shared across all these diseases, whether they share the same expansion-contraction mechanism, but I could imagine they could be very different. I think I saw a poster today targeting a helicase that, when removed, halted the expansion. Was germline transmission? Yeah, I think anything you can imagine. I really can't speak to it, but we're just focused on the outcome. All right, thank you.